Okay, in the morning session we have seen the replication of DNA. In the morning session only we have seen the different types of RNA, because there was a question on the type of RNA. There was a question on this HNRNA. Okay, HNRNA. There was a question on this heterogeneous nuclear RNA. It's a precursor for other RNAs. This was the answer. Okay, we have seen these different types of RNAs. To that we have seen the replication process, different enzymes for the replication. What is the role of DNA polymerase? What is the role of DNA helicase? DNA primase, topoisomerase, DNA ligase, and single-stranded binding protein. So you should know the role of these different different proteins or amino acid proteins and enzymes responsible for the replication process. replication में enzymes होते हैं और इनका क्या क्या रोल है. DNA polymerase. There are three DNA polymerase as far as prokaryotic replication is concerned. So first and second is involved in the DNA repair, or they as exo and endonuclease activity. DNA polymerase 3 is involved in the polymerization of DNA. Then DNA polymerase 3 is also involved in the proofreading activity. DNA helicase is involved in the opening of double-stranded DNA. Primase will synthesize a primer, short RNA primer. Topoisomerase will prevent the supercoiling and mixing activity. DNA ligase will join the Okazaki fragments, and a single-stranded binding protein. They are known as the DNA destabilizing protein. After opening of double-stranded DNA structure by helicase, so these proteins will bind and they will prevent the association. They will prevent the re-annealing of two DNA structure. We have seen this, very important. After that, we have seen the how there is a attachment of this DNA protein, how there is a formation of replication bubble, how there is a formation of origin of replication. After that, proper replication. So in that initiation of replication, RNA primer DNA synthesis, which is a semi-conservative, okay, it is a continuous on leading strand and discontinuous on lagging strand. We have seen that. On the lagging strand, you will find a small fragments of DNA, which is known as the Okazaki fragment. They are joined by DNA polymerase 1 and ligase later on after the replication. Replication, in the replication block, we have seen this role of single-stranded binding protein. After that, we have seen the synthesis occurs in the direction of 5' to 3' DNA polymerase 3. We have seen the role of DNA polymerase 1 and ligase. This is in case of prokaryotes. In case of eukaryotes, there are at least 5 DNA polymerase. The process of replication in the eukaryotes or in the higher animals, that is a little bit complicated. So we have seen the, after that, lastly, replacement of DNA primer by RNA polymerase 1. Sorry, DNA polymerase 1. In eukaryotes, DNA polymerase alpha is there, beta is there, gamma is there, delta is there, and epsilon is there. So we have seen the activities, we have seen the functions of this DNA polymerase. We have seen the replication process in the detail. Now we will see the damage to the DNA, and damage to the DNA or mutations. Okay. First we will see major classes of damage, major type of damage to the DNA. Now replication is the process which is carried out very cautiously in the your cell, and minimum errors are there in a replication. तो replication process होता है वो बहुत ही fidelity से और बहुत ही carefully carry किया जाता है. is DNA is replicated and all the checkpoint has been monitored, but still most of the time there is no damage to the DNA, there is no errors in the DNA while replicating, but sometimes there might be errors in the DNA. Okay, so we'll see errors means mutation, but first damage to the DNA. So for example, if you expose the DNA to the UV light will damage the DNA. Chemicals will damage the DNA. UV light, chemicals, then free radicals, they can also damage the DNA. So there are different physical, chemical methods to damage the DNA. Okay? So first is the deamination, single base alteration. Damage it. This damage may be इधर अल्टर सिंगल बेस है ना, ये डैमेज क्या कर सकता है, जो डीएनए का है उसका एक सिंगल बेस जो है ना वो अल्टर कर सकता है, alteration of single base, right? So for example deamination of one of the either pyrimidine base, pyrimidine base होता है उसमें deamination होगा तो obviously उसका base change होगा, okay, right? Deamination of uracil will convert into another one like that. Then deputination, so deputination means removal of purine thing. Then base alkylation, there is a alkylation of base, insertion or deletion of any one nucleotide and incorporation of base analog. These are the called as single base alteration. Two base alteration, two base alteration, इसमें दो base का alteration हो गया, so either pyrimidine or purine. Well, so UV light induced pyrimidine dimer alteration is a type of two base alteration. Then chain break means oxidative free radical formation and ionizing radiation, they causes the chain break. And cross linkage between the bases of same or opposite strand, cross linking will be there. Cross linking of same base or opposite bases of opposite strand between DNA and protein molecule. That is the different types of damage to the DNA. Actually, this part is not much important for GPAT, but they might ask you these points. Right? That's why I have included. So if you want to read in the details are given in the book. You can read from Biochemistry by Satyanarayana if you are interested. You also can read from the Biochemistry by Harper. Okay. Different type of mutation. What do you mean by mutation? Mutation is any change, any alteration in the native structure of your DNA. Any change or any alteration in the base pair of the native structure of your DNA. Known as a mutation. It may be either point mutation, एक ही जगह पे. It may be either time shift mutation. In the point mutation, it may be transitions or transversions. Transversion में आता है silent mutation, missense mutation, and nonsense mutation. You see it one by one. So mutations means what? Alteration of any base pair in the DNA structure. DNA में कोई base pair change हो गया तो उसको mutation बोलेंगे. डीएनए में कोई भी अल्टरेशन हो है तो वैसे डीएनए में अल्टरेशन बहुत ही कम बार होता है, लेकिन बहुत सारे फैक्टर्स होते हैं जैसे फ्री रेडिकल्स, जैसे पेरोक्साइड, जैसे यूवी लाइट, जैसे आपका अल्कली हो गई, इट कैन कॉज यू म्यूटेशंस. टाइप्स ऑफ म्यूटेशन इज इंपॉर्टेंट फॉर योर जीपैट नाइपर एग्जाम. Mutation is nothing but is a change in the DNA structure. What is mutation? Change in the DNA structure. डीएनए के स्ट्रक्चर में कुछ चेंज आता है तो उसको बोलेंगे म्यूटेशन. चेंज इन द डीएनए स्ट्रक्चर ऑफ ए जीन देन the factor or the agent or the chemicals that can induce a change is known as a mutagen. Any factor or any chemical or anything that can induce a change in the DNA known as a mutagen. Okay? Or the process is known as a mutagenesis. Okay, first is the point mutation. ये थोड़ा सा शॉर्ट में ले लाऊं मैं, इसलिए पहली बार अगर आपने पढ़ा होगा किसी ने उसको ज़्यादा समझ में आएगा. अगर आपने कभी पढ़ा ही नहीं होगा तो आपको थोड़ा सा ऊपर से ही जाएगा, इसलिए थोड़ा सा पढ़ना जरूरी है. जैसे आपने बायोकेमिस्ट्री स्टार्ट किया न्यूक्लिक एसिड, इट्स यूर रिस्पॉन्सिबिलिटी दैट यू शुड एटलीस्ट लुक वंस इन द बुक, है ना. एक बार आपने पढ़ना चाहिए ताकि आपको समझ में आए. कुछ लोग बोलते हैं कि सर समझ में आएगा कैसे, ऑलरेडी आपको एक बार कॉलेज में पढ़ाया है ना, आपने पढ़ना चाहिए. तो आप सिर्फ डिपेंडेंट रहते हो, कॉलेज के ऊपर डिपेंडेंट हो. या फिर मैंने एक बार यहाँ पे एक कॉलेज में गया था, वहाँ पे एक सेकंड ईयर का लड़की थी, उसको पूछा, वो आई मेरे पास, सर मैं कैसे स्टडी करूँ. बोला, मैंने उसको गाइड किया ऐसे ऐसे करो. सर बोली मैंने क्लासेस लगाए. मैंने बोला क्लासेस सेकंड ईयर में क्यों लगाए. नहीं बोले सर, मैंने बोला सर, मैंने बोला इट्स नॉट रिक्वायर्ड इन द सेकंड ईयर. तो बोले सर हमको सेमेस्टर का पढ़ा रहे हो. सर क्या बोली, सेमेस्टर का पढ़ा रहे. फिर सेकंड ईयर में हो आप, कॉलेज में हो, और सेमेस्टर का पढ़ने के लिए भी आप क्लासेस लगा रहे हो. मतलब फिर कॉलेज का रोल क्या है, कॉलेज में पैसे ही भर रहे हो आप, फीस पे कर रहे हो, कॉलेज में क्यों कर रहे हो. तो उनको बोलो सर आप ही एक काम करो, प्रैक्टिकल भी ले लो आप, और क्लासेस में आते हैं हम, तो बाद में डिग्री भी आप दे दो. बचा क्या है. मतलब कहने का मतलब ये है कि इन द सेकंड ईयर इफ यू आर ट्राइंग, इफ यू आर ज्वाइनिंग द क्लासेस फॉर your semester exam and GPAT exam, then what is the role of your college? कॉलेज का क्या रोल है? कॉलेज को सिर्फ आप पैसे क्यों भर रहे हो? तो कॉलेज में आपको पढ़ाते हैं, कॉलेज में पढ़ना नहीं सर, कॉलेज में भी पढ़ाते हैं लेकिन मतलब बस फ्रेंड ने लगाया, अपने लगाया. तो point mutation means replacement of one of the base pair by another is nothing but the point mutation. आपको as it is question आ सकता है. What is point mutation? Replacement of one base pair with another one is known as a point mutation. Okay. There are two types of this point mutation. First is the transitions. In this case, either purine base जो होता है or pyrimidine, they are replaced by one another. Either purine is replaced by pyrimidine, purine is replaced by purine. Known as a transition. There is a transition होता है. Transposition means replacement of purine by pyrimidine और pyrimidine by purine. बोलेंगे सर दोनों में डिफरेंस क्या है? इसमें रिप्लेस हो सकता है, इसमें प्यूरिन रिप्लेस होगा, है ना, pyrimidine से, pyrimidine रिप्लेस होगा प्यूरिन से. इसमें वैसा नहीं है. यहाँ पे प्यूरिन विद pyrimidine एंड वाइस वर्सा. ट्रांसवर्जन ट्रांजिशन मतलब क्या? एक ही जगह पे, मतलब यहाँ पे से, है ना, if there are three base pair adenine, guanine, thymine, okay, on single strand, if it is altered known as a transition. दूसरे एक सिंगल स्टैंड पे एडेनिन गुआनिन थाइमिन की वजह यहाँ पे एक अल्टर होगा, है ना, दैट इज अ ट्रांजिशन. ट्रांसफॉर्मेशन मींस व्हाट? दोनों स्टैंड में कॉन्सिक्वेंस ऑफ पॉइंट म्यूटेशन. इसका म्यूटेशन के क्या हो सकता है? पॉइंट कॉन्सिक्वेंस ऑफ पॉइंट म्यूटेशन, साइलेंट म्यूटेशन. साइलेंट म्यूटेशन मींस द कोडन ऑन mRNA containing charged base may code for same amino acid. चार्ज नहीं, चेंज्ड. द कोडन ऑन mRNA के ऊपर जो कोडन है जैसे UAG, सो कंटेनिंग ए चेंज्ड बेस. UAG अगर चेंज हो गया, UAC हो गया, सो UAG will code for one amino acid and UAC will also code for amino acid. And if those two codes for same amino acid, one is a silent mutation. अगर mRNA में कोडन में अगर एक बेस अल्टर हो गया, अल्टर होने के बाद में भी अगर वो सेम ही अमीनो एसिड को कोड कर रहा है तो उसको बोलेंगे साइलेंट म्यूटेशन. द कोडन ऑन mRNA containing changed base may code for same amino acid. For example, UCA will code for serine. UCA कोड जो होता है mRNA के ऊपर, ये कोड करता है सेरिन के लिए, है ना, यूरासिल साइटोसिन एंड एडेनिन. UCA will code for serine amino acid. Now, if it is altered, an altered code UCU, एडेनिन की जगह यहाँ पे यूरेसिल होगा, UCU. अगर ऐसा चेंज हो गई, वो भी कोड करता है सेरिन के लिए. So this defect is not detectable in the mRNA, and that's why it is known as a silent mutation. Now, missense mutation. Missense mutation means change the base may code for different amino acid. So in this case, UCA will code for serine and UCU also code for serine. But if UCA code for serine, UCU code for another amino acid, it is a missense mutation. Change in the base may code for different amino acid. Like for example, UCA will code for serine, altered ACA. से U की जगह यहाँ पे फर्स्ट बेस पेयर चेंज होगा. ACA will code for threonine. So, okay, UCA code for classical example of such kind of missense mutation. किस में होता है missense mutation? In case of sickle cell anemia. सिकल सेल एनीमिया के जो पेशेंट होते हैं उसमें ये म्यूटेशन होता है. कौन सा? missense mutation. In the sickle cell anemia, this type of mutation is there. Now it is confirmed. Okay. Frame shift mutation. Frame shift mutation is particular pair occurs when one or more base pair either is inserted in the sequence or deleted from the DNA sequence. Okay. It occurs frame shift mutation occurs when one or more base pair either inserted or deleted from the DNA, causing the either insertion or deletion mutation, respectively, known as a frame shift mutation. Now, nonsense mutation, because of the altered base may become termination. because of the alteration of base, अगर कोई alteration हो गया जैसे UCA की जगह UGA बन गया, तो UGA क्या है? टर्मिनेशन कोड है. एग्जांपल UCA है ना, UCA will code for serine, and if alteration is there, it may become UAA. अगर ये C वाला A आ गया, that is known as a nonsense codon or termination codon. This kind of mutation is known as a nonsense mutation. This kind of mutation is known as a nonsense mutation. So these are the different mutation types. Very important question आता है इसके ऊपर. So missense mutation, silent mutation क्या होता है, missense mutation क्या होता है, mutation क्या होता है, frame shift mutation क्या होता है, nonsense mutation क्या होता है, इसके ऊपर क्वेश्चन आता ही आता है. Now next one is okay, cell cycle. Now when replication will occur, your cell has a particular lifespan. Your cell will divide by the process of mitosis. Your cell will divide by the process of mitosis. Mitosis and meiosis, they are the process for cell division. Now when cell will division, cell division के different phases होते हैं, known as a cell cycle. Cell cycle of mammalian cell. Okay, so cell cycle of mammalian cell will have different cell. Okay, different different stages. So G0, this is for example G0 phase, G0 phase. So in this phase, 0 means it is a resting phase of your cell cycle. But in this phase, cell will prepare itself for the mitosis. In this phase, it is a resting phase, but cell will prepare itself for the mitosis. इसमें क्या होगा, G0 phase में जितने भी एंजाइम्स लग रहे हैं अपने सेल के लिए मल्टीप्लाई होने के लिए, जितने भी प्रोटीन्स लग रहे हैं, जितने भी जो भी लग रहा है, मतलब रॉ मटेरियल यहाँ से प्रिपेयर करेंगे. देन G1 फेज में अगेन प्रिपरेशन आएगा, प्री प्रिपरेशन. G0 is the resting phase, G1 phase is the preparation. Now S phase is nothing but the synthetic phase. S phase is the synthetic phase. The synthetic phase means what? In this synthetic phase, so there is a replication, there is a transcription, and there is a S phase, that is known as synthetic phase. इसमें ही आपका DNA का replication होता है, इसमें ही आपका DNA का transcription होता है, translation होता है, right? So this is a cell cycle, and this is a programmed cell cycle. Okay? And then G2 phase. G2 phase means whatever DNA is replicated, whatever RNA is made, whatever protein is synthesized. So that whether that protein, RNA, DNA, whether that is correct or not, that is checked in the G2 phase. So that is corrected in the G2 phase. Okay, and then it will go on the M phase. M phase means mitotic phase. So whatever DNA is synthesized, whatever DNA is corrected, whatever proteins are synthesized, that will be assembled as a mitotic spindle. और जहाँ पे आपका जो जीन होता है वो डिप्लॉयड होता है. जीन मीन्स इट इज अ डिप्लॉयड in humans. So this diploid gene is assembled and converted into quadruploid. यहाँ पे बनता है quadruploid. इसमें mitotic phase में, and then वहाँ पे spindle बनता है. Okay, divided into two spindles. Okay? Two spindles of each diploid, and then there is a development of two different cells. Most of the anti-cancer drugs. Okay, anti-cancer means what? Cancer means controlled proliferation of cell, uncontrolled growth of cell. Okay, so if uncontrolled growth is there is a cancer, right? Now most of the drugs, they will act on this cell cycle space, known as the phase specific. So most of the anti-metabolite, alkylating agent, and anti-metabolite alkylating agent, they will act on a S phase. Because active synthesis of material takes place in the S phase. Some will act on G1 phase and G2 phase. Right? That we'll see. that we'll see in a anti-cancer drugs. Okay, but यहाँ पे अगर anything is damaged, if faulty DNA or faulty RNA is synthesized, okay, so it is recognized and detected. Okay, detection of incomplete replication or replicated DNA. Now whatever damage is there, so that is detected in the G2 phase. So in the M phase, improper spindle formation, that is detected. Okay, now in case of damaged DNA, that is detected in the G1 cell. Now, if damage is there and if it is incorrectable, so cell will try to correct itself by different mechanism. correction mechanisms are also there. So if there is a damage to DNA, if there is altered DNA, if there is a faulty DNA, so it will be corrected by cell itself. If it is not correctable, अगर correct नहीं हो रहा है, so cell will be sent to a apoptosis. Apoptosis is nothing but the programmed cell death. So if damage to the genetic material is not correctable, then it will send to the apoptosis. It is cell programmed cell death. This is a cell cycle. After the replication, the things occurs in the S space. Replication, transcription, ये सब इसमें होता है. Now we'll see the next part, that is a transcription part. Transcription. So what is meant by genome? Genome means what? Genomics. Genomics or genome. Genome means your DNA. Structural structural genomics refers to a structural modifies and complete protein structure. There are three terms: genome, transcriptome, and proteome. Proteome. Genome refers to the genetic material, that is DNA, RNA. Okay, comparative genomics involves the study of comparative gene and functional phylogeny. Metagenomics refers to the genome and microorganism, genes of microorganism. Metagenomics. Proteome is nothing but the protein storage of cell. Repertory, that is a source of protein with their neural and biological function. Now, what do you mean by transcription? Transcription is nothing but the process of synthesis of RNA from the DNA. It is the process in which RNA is synthesized from DNA. Okay. Transcription. We have seen the central dogma of life. So, replication of DNA, formation of identical copies of DNA is known as replication. Then from DNA, synthesis of RNA is there, is known as transcription. And from RNA, there is a synthesis of protein known as a translation. That is the central dogma of molecular biology or central dogma of your life. Okay. The process in which RNA is synthesized from DNA. Okay. DNA is DNA consists of genetic information. Genetic information is there in the DNA. Now, which is expressed in the RNA, known as a transcription. The product formed in the transcription is known as a primary transcript and subject to the post-transcriptional modification to produce the finally active RNA molecules. तो DNA से क्या बनता है? RNA बनता है. कौन सी प्रोसेस है भाई? ट्रांसक्रिप्शन. नाउ प्राइमरी जो बना है ट्रांसक्रिप्ट, इससे ट्रांसक्रिप्शन कंप्लीट होने के बाद में पहले जो मिलता है आपको वो है प्राइमरी ट्रांसक्रिप्ट एंड इट इज सब्जेक्टेड टू पोस्ट ट्रांसक्रिप्शनल मॉडिफिकेशन. तो उसमें चेंजेस होता है, जैसे निक्सेलिंग, एडिशन ऑफ पॉली ए टेल, एडिशन ऑफ एडिशन डिलीशन ऑफ समथिंग, known as a post transcriptional modification to produce a finally active RNA molecule. We have seen the formation of identical copy of DNA is known as a replication. From DNA, formation of RNA is nothing but the transcription. From RNA, okay, expression of DNA in the RNA is known as a transcription, and from the RNA, synthesis of protein is known as a translation. It is central dogma of life or central dogma of molecular biology. The overview of your RNA, this is not transcription. Okay, so we have seen in the DNA polymerase, we have seen the replication, DNA polymerase. So DNA polymerase is there. There is a type of DNA polymerase. In that you require a primer and etc etc. This is not the case in transcription. Transcription में आपको यहाँ पे primer, यहाँ पे DNA polymerase के type वगैरह इतना सारा मैटर नहीं. Okay, यहाँ पे सिर्फ क्या है? DNA polymerase is the only enzyme known as a DNA polymerase. It will attach to a DNA. DNA polymerase will attach to a DNA at a particular sequence. At a particular sequence. DNA में कुछ sequence होगा, जैसे TAT, टाटा बॉक्स होता है या प्रिंगो बॉक्स होता है ना. So on the DNA there is a promoter region. It is known as a promoter region. So there is a particular sequence of base pair. DNA के ऊपर एक पर्टिकुलर बेस पेयर के सीक्वेंस हो गए, जैसे ATA टाटा बॉक्स, TATA टाटा बॉक्स या फिर प्रिंगो बॉक्स या फिर कुछ 35 बेस पेयर का एक सीक्वेंस होता है ना. अब वो है ना, वो एक sequence. So such kind of sequence they are known as a promoters. So this RNA polymerase will attach to a promoter region and then it will cause initiation of transcription. Then it will form a RNA. Initiation, then elongation is another step, then termination is the another step. So binding of DNA polymerase to the binding of RNA polymerase to the DNA is a very important prerequisite for this transcription process. बाद में initiation हो जाएगा, बाद में elongation, दूसरा step, तीसरा termination, and बाद में mRNA, synthesized mRNA will be released. After that, there is a post-transcriptional modification. So this is just overview. ये सब steps है, अपन detail में देखेंगे. So the process of transcription. So out in the different step. First was the initiation. Initiation means what? Binding. Here is only one RNA polymerase. So binding of RNA polymerase to the DNA is important step for transcription. The enzyme binds to a specific region known as a promoter region. ये जो DNA, ये जो RNA polymerase enzyme है, जो DNA के ऊपर bind होगा, RNA polymerase will bind to a DNA at a particular sequence known as a promoter region. There are two base pair sequences on the coding DNA strand. तो DNA में दो स्टैंड होता है, एक को यहाँ पे बोलते हैं कोडिंग स्टैंड और एक को बोलते हैं नॉन कोडिंग स्टैंड. और कोडिंग स्टैंड जो है उसको बोलते हैं सेंस स्टैंड, जो कोडिंग है उसको बोलते हैं नॉन सेंस स्टैंड. दिस DNA polymer, this RNA polymerase as a RNA polymerase जो है ना, this एंजाइम RNA polymerase, उसमें यूनिट सब यूनिट होता है. फॉर एग्जांपल जैसे 2 अल्फा सब यूनिट होता है ना, 1 बीटा डैश होता है, 1 बीटा सब यूनिट, और एक कोफैक्टर होता है उसको बोलते हैं सिग्मा फैक्टर. नाउ इट इज अ RNA polymerase of E. coli. So RNA polymerase of eukaryotes is different. Say for example RNA polymerase of E. coli. So there is alpha, 2 alpha subunit, 1 beta subunit, and 1 beta dash subunit, and 1 sigma factor. When this enzyme will attach to a DNA at a promoter region. So two way, okay, promoter region means particular sequence. Okay, which sequence this? Pinbow box. Pinbow box. So it is also known as a Tata box. यहाँ पे there is a particular sequence, nucleotide sequence on the DNA, TATAT. Now if such a sequence is present, there is known as a Pinbow box, is a promoter region. तो जो RNA polymerase है वो east side पे अटैच होगा DNA में, है ना. So Tata box is made up from 6 nucleotide bases, that is TATAAT. Okay? That's why known as a Tata box, TATAA. So add this nucleotide sequence which is located near to the starting point of transcription. Tata box is located at the near point of near starting point to the transcription. Another promoter region is there known as a 35 sequence. Second recognition site for promoter region of the DNA. ये sequence होता है TAGAC, located about 35 bases upstream from the site of transcription. तो ये जो एंजाइम है RNA polymerase, which is alpha, 2 alpha subunit and beta dash, and then sigma as a cofactor. Attachment of this RNA polymerase to a DNA at a particular base sequence, that is a Pringler box or 35 sequence, that is a promoter region. Now the sigma factor of DNA polymerase, sigma factor of RNA polymerase will recognize and initiation and for the initiation of ट्रांसक्रिप्शन. मतलब क्या कि आरएनए पॉलीमरेज डीएनए जो स्ट्रक्चर है डबल स्ट्रैंडेड डीएनए उसको बाइंड करता है. कहाँ पे बाइंड करता है? प्रोमोटर रीजन पे बाइंड करता है. प्रोमोटर रीजन क्या होता है? ये पर्टिकुलर बेस पेयर का सीक्वेंस होता है. कौन सा? इधर ये होता है टाटा बॉक्स जो नियर होता है ट्रांसक्रिप्शन स्टार्टिंग पॉइंट से, या फिर आपका दूसरा 35 सीक्वेंस होता है जैसे TTGAC. तो इट इज ऑल्सो लोकेटेड अबाउट 35 बेस पेयर अपस्ट्रीम फ्रॉम द साइड ऑफ ट्रांसक्रिप्शन. तो यहाँ पे ये RNA पॉलीमरेज डीएनए को इस साइड, ये जो साइड है डीएनए के ऊपर होता है, So these sequences, they are present on the DNA, and to this sequence RNA polymerase will bind. RNA polymerase में क्या है कि it has a structure like this. उसमें there are two alpha subunit, there are beta dash, and there is a sigma. और this sigma factor will recognize the promoter region. sigma factor होता है एंजाइम में, वो promoter region को ढूंढता है DNA में, तो वहाँ पे जाके अटैच होता है. And then there is the initiation of transcription. Next process is nothing but the elongation. Next process is nothing but the elongation. So a holo enzyme RNA polymerase recognize the promoter region. Holo enzyme of there is a holo enzyme is active part. Enzyme plus prosthetic group. The prosthetic group होता है sigma factor. So it recognize the promoter region. The sigma factor is released and transcription will be proceed. Now RNA is synthesized from again 5' to 3' direction. RNA is synthesized from 5' to 3' direction, which is parallel to the DNA template. The DNA जो होता है 3' to 5' end, तो उसके anti parallel ये 5' to 3' by the RNA polymerase. DNA is synthesized in a direction of 5' to 3', which is anti parallel to the DNA template by RNA polymerase. Now RNA polymerase will use these nucleotide bases: adenine triphosphate, guanine triphosphate, cytosine triphosphate, and uryl triphosphate. These base pairs they are used because in the DNA you have thymine and in the RNA you have uracil. For the formation of RNA, upon addition of each neutral trait of growing chain, pyrophosphate moiety is released. एक एडिशन में नाउ एडिशन ऑफ दिस बेस पेयर इज अ कॉम्प्लीमेंट्री. जैसे डीएनए में अगर थायमिन होगा तो यहाँ पे यूरासिल आएगा. डीएनए में अगर थायमिन होगा तो यहाँ पे यूरासिल आएगा. एडेनिन गोनिन थायमिन साइटोसिन यूरासिल. तो पिरिमिडिन्स आर 3. तो जो पिरिमिडिन्स है इंस्टेड ऑफ थायमिन यहाँ पे यूरासिल आएगा. These four traits are used by RNA polymerase to synthesize the RNA. Now, RNA polymerase does not require any primer. RNA polymerase does not require any primer and do not possesses any exo or endonucleases activity. So DNA polymerase में था. DNA polymerase के लिए एक primer लगता था, RNA primer. उसके बाद में DNA polymerase has a proofreading activity and also exonucleases, endonuclease activity. So such activity is not there in case of. Due to lack of proofreading activity, the RNA polymerase does not have ability to repair the mistakes in the RNA synthesis. So DNA synthesis में कुछ mistake होगा तो DNA repair करता था उसको, DNA polymerase. But RNA polymerase does not have proofreading activity. That's why it cannot repair the mistakes in the RNA synthesis. But mistake seldomly occurs. Now termination of RNA synthesis. Termination of transcription. Transcription process stops by termination signal, which are of two type. टर्मिनेशन कब होगा? जब ये टर्मिनेशन सिग्नल आएंगे. कौन से भाई? पहला है आपको रो डिपेंडेंट टर्मिनेशन, सेकंड होता है रो इंडिपेंडेंट टर्मिनेशन. सो व्हाट डू मीन द रो डिपेंडेंट टर्मिनेशन? इट इज अ पर्टिकुलर प्रोटीन, स्पेसिफिक प्रोटीन रो फैक्टर. It is a particular protein rho factor which binds to a growing chain of RNA and also partly bind to the DNA and terminates the process. Okay, terminates the process. Okay, terminates the process. Also responsible for the dissociation of RNA polymerase from the DNA. DNA से RNA polymerase को भी हटा देगा. That is the row dependent termination. एक specific row factor होता है. Now row independent termination. So formation of hairpins of newly synthesized RNA termination is brought out. This occurs due to the palindrome. Palindrome मतलब क्या? पर्टिकुलर सीक्वेंस होता है. पैलिंड्रोमिक सीक्वेंस. जैसे मैंने अगर यहाँ पे बोला है रोटर वर्ड लिखा ROTOR. There is one word known as rotor, ROTOR. So if you read from right side to the left side, rotor. If you read from left side to the right side, again ROTOR. रोटर ROTOR and ROTOR. आप कैसे भी पढ़ो, राइट साइड से पढ़ो तो रोटर ही प्रोनाउंस होगा, लेफ्ट साइड से पढ़ो रोटर ही प्रोनाउंस होगा. और एक वर्ड बोलता हूँ मैं, मैडम, MADAM. राइट साइड से पढ़ेंगे तो भी मैडम ही दिखेगा आपको, लेफ्ट साइड से पढ़ेंगे तो भी मैडम ही दिखेगा. तो सच काइंड ऑफ सीक्वेंस इज नोन एज अ पैलिंड्रोमिक सीक्वेंस. Such kind of palindromic sequence. रो independent termination is brought about by palindromic sequence, palindromes. रोटर हो गया, मैडम हो गया, जैसे ATG इज अ सीक्वेंस होता है, पर्टिकुलर सीक्वेंस. रोटर जैसे ROTOR, यहाँ से पढ़ेंगे ROTOR, यहाँ से उल्टा पढ़ेंगे ROTOR. उल्टा पढ़ेंगे है ना तो भी रोटर दिखेगा, सीधा पढ़ेंगे तो भी रोटर दिखेगा. जैसे मैडम वर्ड लिखेंगे अभी आप मैडम, सीधा पढ़ेंगे तो भी मैडम ही दिखेगा, उल्टा पढ़ेंगे तो भी मैडम ही दिखेगा. that is known as a palindrome or palindromic sequence. Such kind of palindromic sequence is present in the particular site on the DNA. तब तक चलता रहता है, जब palindromic sequence आएगा तब वो synthesis. So first step होता है, first step is nothing but the binding of RNA polymerase to the DNA. So this is the RNA polymerase. This will bind to a particular base pair sequence. फॉर एग्जाम्पल ये होता है क्या, 35 पिन बो स्टार्टिंग पॉइंट, यहाँ पे कुछ सीक्वेंस रहेगा ATGC. RNA polymerase will bind to that sequence known as a promoter region. इसको बोलते हैं अपन promoter region. So RNA polymerase will bind to a promoter region. Sigma factor will be dissociated. Sigma factor will recognize that promoter region and will bind to that. Okay? So sigma factor will be dissociated. Then there is a synthesis of mRNA according to the parent DNA structure. One strand is known as a coding strand, one is known as a non-coding strand. Coding strand is known as a sense strand, and non-coding strand is known as a non-sense strand.
Hello sir, namaste. First advisory.
कहाँ से आते हैं ये? Okay. Now transcription process summarized over here. Okay. The core enzyme means that is nothing but the main enzyme. Okay. Then sigma factor, sigma subunit. So that is known as a holoenzyme. Core enzyme plus sigma subunit known as a holoenzyme. ना आपने देखा है holoenzyme means protein part plus non-protein part or a cofactor. It is active form of enzyme known as a holoenzyme. So these two together, core enzyme plus sigma factor known as a holoenzyme. Now it will recognize a particular sequence on the double-stranded DNA. Sigma factor will recognize. Then there is a binding of this DNA RNA polymerase on a DNA. Particular point known as a promoter region and initiation point. Okay. Now initiation चलेगा यहाँ से up to this point, ये termination point तक वो copy करता रहेगा DNA के एक strand को. So DNA, one strand of DNA, that is a coding strand, will be copied complementary by the DNA RNA polymerase. So it will synthesize the RNA polymerase. So first step is initiation. Then second step is this is the first step, initiation. Then second step, chain elongation. And third step is the termination. Okay, and you will get your mRNA. We'll see this again. Okay. So what do you mean by palindromic sequence? So this is known as a DNA. ये आपका DNA है, double stranded DNA. One is known as a coding strand, and one is known as a template strand. So on a DNA you will have a particular sequence known as a promoter region. यहाँ पे एक particular sequence रहेगा. जैसे for example यहाँ पे TA data box और print now box और data box. Then another region is there known as 35 sequence. यहाँ पे भी एक region है, right? So this region is known as a promoter region, and this region is known as a coding region for gene. So this region will be copied by mRNA. ये जो region है coding region for gene, this region will be copied complementary on a mRNA. For example, जैसे DNA में जैसे coding strand है, इसके टेम्पलेट स्टैंड है, तो इसके कॉम्प्लीमेंट्री क्या बनेगा, आरएनए बनेगा. जैसे डीएनए में अगर थायमिन है तो यहाँ पे एडेनिन आएगा, उसके अपोजिट में एटी आता ना, ATGC ना, ए बॉन्ड विथ T, adenine thymine guanine cytosine, ऐसा होता है adenine thymine guanine cytosine चार ऑप्शन। देन अगर यहाँ पे थायमिन है तो आरएनए में थायमिन नहीं रहता है, यूरेसिल आएगा। ए के अपोजिट टी तो इसमें आएगा नहीं आरएनए में, तो इसमें यू आता है। जी के अपोजिट जी, सी के अपोजिट जी आएगा, टी के अपोजिट सी आएगा, टी के अपोजिट ए आएगा, है ना। अगेन इसमें टी आना चाहिए, टी की जगह यू आएगा, साइटोसिन का अपोजिट कौन आएगा। ये ऐसा कॉम्प्लीमेंट्री स्टैंड इट इज सिंथेसाइज्ड। ट्रांसक्रिप्शन कॉम्प्लीमेंट्री बेस पेयर रिलेशनशिप, उसकी कॉम्प्लीमेंट्री बेस पेयर रिलेशनशिप you can see over here the transcription process again. So this is a DNA, आपका DNA होता है डबल स्ट्रैंडेड DNA। on the DNA there is a promoter region or promoter site, then transcription unit means this is copied and another termination site is there, termination site is there. Now what happens, so this is known as a RNA polymerase, this is known as RNA polymerase, okay, and its active enzyme known as a holo enzyme along with the sigma factor, RNA polymerase, it will bind to a promoter region, at a promoter region जिससे bind होगा, sigma factor will recognize and it will bind to the promoter region, then sigma factor will be released, it will start the transcription known as the initiation of transcription. Now, elongation process क्या होगा, so RNA polymerase will do its work, it will copy the coding strand complementary on, so like this new RNA will be formed, elongation will be there। कब तक elongation होगा, जब तक आपका termination sequence नहीं आएगा, termination site नहीं आएगा। so when termination sequence will come, okay, so elongation will stop, okay, elongation will stop। so like for example termination sequence आ गया अभी, palindromic sequence आ गया, so either it is a row dependent termination or row independent termination, है ना। termination point में row attach होगा, row factor will attach to a termination site, it will release the RNA polymerase, it will also release the newly synthesized mRNA. Otherwise here is some palindromic sequence and that is known as row independent termination, so you will get a newly synthesized RNA, newly synthesized RNA known as a transcription, synthesis of RNA from the template, that is a transcription. Are you getting or not? Now what do you mean by genetic code? Genetic code, so genetic code means, so there is a three nucleotide base sequences, three nucleotide base sequences in the mRNA, which is a code word. Well, now three nucleotide base sequences, so where from it come, they came from DNA only, जैसे DNA के कोडिंग कोडिंग स्ट्रक्चर में या फिर टेबलेट स्ट्रक्चर में जो कोडिंग स्टैंड जो होता है डीएनए का, उसके अपोजिट और उसके कॉम्प्लीमेंट्री ही आपका एमआरएनए बनता है। जैसे कोडिंग स्टैंड के ऊपर अगर डीएनए के एडेनिन होगा, है ना, एडेनिन होगा, तो कॉम्प्लीमेंट्री एमआरएनए के ऊपर क्या आना चाहिए, है ना, एडेनिन गुआनिन आएगा, है ना। उसके ऊपर अगर साइटोसिन होगा तो आना चाहिए थाइमिन, लेकिन थाइमिन की जगह यहाँ पे यूरेसिल आएगा। सो वट एवर कोडिंग इज, वट एवर बेस पेयर इज प्रेजेंट ऑन द डीएनए, so same thing complementary base pair are added to synthesize the RNA molecule, okay. So three nucleotide base sequence on mRNA is a code word for amino acid, is a code word for amino acid, which is nothing but the genetic code. Simply they are also known as simply codons, okay. There are only 20 amino acids, there are possible 64 codons, there are 64 codons possible combination and only 20 amino acids, 64 codon will code only 20. The dictionary the dictionary of nucleotide bases, that is adenine guanine cytosine uracil, that will decide the sequence of amino acid, that will decide the sequence of amino acid in the protein. So whatever codes present on the mRNA के ऊपर sequence क्या है, the sequence of mRNA will decide a sequence of amino acid in the protein. Whatever codes are on a mRNA, that is read, and based upon that, okay, the codon 3 base pair codon will have one will code for amino acid. They will code for one amino acid. Okay, let's say for example, the three codes UJ and UGA, UAA, AG and UGA, known as amber, ochre, and opal, respectively. They do not code for any amino acid. They do not code for any amino acid, and that's why they are known as a termination codons or stop codon. ये तीन जो है ना आपको याद AA, UGA and UGA, amber, orchard or opal. These are known as, they will not code for some amino acid. They are known as a stop codon or nonsense codon. The nucleotide sequence on mRNA is read from 5' to 3' end. The codon, for example, codon UAG and use okay, CUG is known as initiation codon. UAG and CUG are known as initiation codon. Now features of genetic codon. ये क्या है? Universality. Same codon will code same amino acid in the all living organism. मतलब क्या? कोई अमीनो एसिड पर्टिकुलर एक कोड है ना, तो वो पर्टिकुलर अमीनो एसिड को कोड करता है. जैसे फॉर एग्जांपल जैसे कोई भी अमीनो कोड होता है 61 कोडन में से पर्टिकुलर कोड करेगा. जैसे ये इनिशिएशन कोडन जो होता है ना, AUG will code for phenylalanine. So AUG will code for phenylalanine in everyone ना. मतलब ह्यूमंस में भी AUG अगर कोड है तो phenylalanine का कोड करेगा. If it is found in the plant, same thing will be coded. If it is in the animals, in the microbes, same will be coded. AUG will code for phenylalanine in everyone. Okay? So that is the universality. Same codon will or same codon codes for same amount all living organisms. But few exceptions are there. So UAA, U, sorry, AUA, AUA code will code for methionine amino acid in mitochondria. So AUA codon will code methionine in the mitochondria, but will code isoleucine in the cytoplasm. Specificity, they are very specific. Specific code will code a particular amino acid. One amino acid will be coded by multiple codons, non-overlapping. It is the feature of genetic code. Code is read from fixed point as a continuous base sequence. Addition or deletion of one or more bases will be change the message sequence on mRNA, called as a mutation. अगर कोई बेस अल्टरेशन हो गया तो क्या बोलते हैं उसको म्यूटेशन बोलते हैं. The genetic code is a degenerate. Most of the amino acid most of the amino acid will have more than one codon. As there are 61 codons and only 21 amino acids are available. Codons on mRNA will recognize a anticodon on RNA. जो mRNA के ऊपर जो 3 सीक्वेंस है न्यूक्लियोटाइड codon that will be read by rRNA है ना, राइबोसोमल RNA के ऊपर एंटीकोडन रहेंगे इसके. तो इनिशियली RNA एक ही बनता है mRNA, उसके बाद में उसका मॉडिफाई करके tRNA बनते हैं, उसके बाद में मॉडिफाई करके उसका rRNA बनता है. What is Oval hypothesis? Oval hypothesis was given by Crick. The tRNA will recognize more than one codon due to third base at a 3' sequence. This explains the detergency of the genetic code, and that is the existence of multiple codons for a single amino acid. देखा था मैं. It is a oval hypothesis. So for example, genetic code along with the respective amino acids. First base at a 5' end. तो ये mRNA के ऊपर आप 5' end से रीड करते हो. पहला बेस आता है अगर U, यूरिडीन, यूरासिल आता है, तो there are 3 base pair, there are 3 bases ना. यूरासिल अगर आता है और यूरासिल आता है, कॉम्बिनेशन क्या होता है? ये तो 2 बेस पेयर हो गए, लेकिन कोडन में 3 आता है. तीसरा कौन सा है? तीसरा भी यूरासिल लग रहा है. U if 3 base pair, for example, one base of 5' end, then middle base is also U, second middle base, अगर तीनों आता है U, so it will code for phenylalanine. और है ना, अगर U आता है, it will code for phenylalanine. UA आता है, that will code for leucine. UUG आता है, मतलब एक ही लेना है, that will call for also leucine. So in this similar way, this UCC, UGA, UGG will call for serine amino acid. likewise UA combination permutation and combination, तो ये कोड भी क्या है ट्रिप्टोफैन को कोड करेगा UAA and UGA. UAG is a stop or nonsense codon. UGA is also stop codon. बाकी कोडन जैसे मतलब ये यहाँ से U इनिशिएशन ये कोडन है पहले 5 डेज के 3 डेज के ऊपर ये कोडन है से है ना C लेंगे तो है ना बीच में सेकंड पेज में भी आपको 4 कोडन है तो ऐसा 61 codons आते हैं. So likewise that if first codon on the permutation is C, so different combinations available like this. So these codons will code only using amino. These 4 codons will code only this proline amino acid. These codons, 2 codons will cause histidine, code histidine amino acids. This will codon glycine like this will these 4 will code only arginine amino acid. Like this, अगर फर्स्ट बेस आता है एडेनिन ना थर्ड बेस आता है यूरेसिल बीच में जो सेकंड बेस है इसमें से चारों भी ट्राई करना जैसे यू ए यू जी ए यू ए विल कोड फॉर आइसोल्यूसिन एंड ए यू जी विल कोड फॉर मिथियोन. दिस इज नथिंग बट द इनिशिएशन कोड. ए यू जी होता है ना तो फर्स्ट अमीनो एसिड ओके फर्स्ट अमीनो एसिड कैरिड बाय टीआरएनए टू द protein synthesis is always methionine. Is always methionine. First amino acid carried to a protein synthesis is always methionine. AUG will serve as a initiation codon. And बाकी तो UAA, UGA, UQA, UGA and UAG they are known as a termination codon. This is known as genetic code. आपको ये पूरा पढ़ के बोलने में कोई मजा नहीं है. Now we'll start